Hello, everyone. My name is Ying Ma. I'll introduce the paper submitted to iMeta. The title is The Prognostic and Therapeutic Significance of Polyunsaturated Fatty Acid-Derived Oxylipins in ST segment elevation myocardial infarction. The recurrent major adverse cardiovascular event MACE rates after ST segment elevation myocardial infarction STEMI remain high. Polyunsaturated fatty acid PUFA-derived oxylipins regulate systemic inflammation and exert cardiovascular effects. However, limited clinical data exist on the prognostic value of oxylipins in the STEMI population. We aim to investigate the association of oxylipin profiles with recurrent MACE, the roles of the key oxylipin predictors. In this study, we constructed an oxylipin-based prediction model that showed powerful performance in predicting recurrent MACE in two independent cohort of 1046 patients with ST segment elevation myocardial infarction. We also demonstrated that 6 anti-inflammatory pro-resolving oxylipin combination had synergistic and cardioprotective effects against myocardial ischemia reperfusion injury. A total of 645 subjects with STEMI who were enrolled at Beijing Anshan Hospital were included in the discovery cohort. Another 401 individuals from the Peking University Third Hospital-built external cohort were included as the independent validation set. Then, targeted metabolomics and machine learning algorithms were performed to develop an oxylipin-based risk model to accurately predict recurrent MACEs after STEMI with 2 years of follow-up. By using targeted metabolomics, we determined the plasma levels of 7 prototype PUFA precursors and 130 species of oxylipin products, metabolized by cyclooxygenases, lipoxygenases, and cytochrome P450. In the discovery cohort, the differentiated oxylipins between the patients with and without recurrent MACEs were mainly derived from arachidonic acid, eicosapentaenoic acid, and linoleic acid, included 9 subclasses. An optimal marker panel consisting of 14 oxylipins that showed significant performance in predicting recurrent adverse cardiovascular events with significant predictive accuracy. The predictive accuracy of the 14 oxylipin panel-based risk model in the external validation cohort was also highlighted, with a significantly accurate prediction rate. Top 6 anti-inflammatory pro-resolving oxylipin ARO combination showed better predictive performances than the other ARO combinations and the pro-inflammatory oxylipin combination. Furthermore, the plasma levels of 6 oxylipins in healthy individuals were higher than those in patients with and without recurrent MACEs, suggesting that those oxylipins might be consumed during the progressive phases of STEMI. Our results demonstrated that the pro-inflammatory oxylipin combination did not alter myocardial injury in myocardial ischemia reperfusion model mice, whereas the anti-inflammatory pro-resolving oxylipin ARO combination significantly protected against MIR-induced abnormalities and injury. The treatment of ARO combination also significantly elevated the plasma and myocardial concentrations of anti-inflammatory and pro-resolving oxylipin. The treatment of anti-inflammatory and pro-resolving oxylipin combination also significantly inhibited the ceramide and lysophospholipid synthase, and ameliorated their downstream pro-inflammatory signaling, including the nuclear factor kappa B activation and nitric oxide synthase. In conclusion, the present study demonstrated that a variety of PUFA-derived oxylipin metabolites serve as residual risk markers for prognosis after STEMI, and the oxylipin-based risk model exhibited powerful performance in predicting recurrent MACE. Furthermore, this study opens a new range of possibilities for the design of bioactive oxylipin agents as intervention points to mitigate post-MIR pathogenesis with significant therapeutic potential for improving adverse clinical outcomes after STEMI.